Let's finish off the respiratory system. We're going to go into quite a lot of detail about external respiration. We're going to lightly touch on internal respiration again. And then actually we're going to go into pulmonary ventilation. We're going to talk about Boyle's law briefly and talking about the muscles of inspiration and expiration, the brain centers involved in the movement of those muscles, and then also inputs into those brain centers. Well, because I feel like kind of rewatching that last video, I felt like, boy, it just gets to be really detailed when we talk about internal and external respiration, and I thought just a simple repeat of all the information might be a good idea. So external respiration happens at the level of the alveolus, the air sac of the lungs. Internal respiration happens in any vascular tissue of your body, and some hypothetical tissue here. We'll say some lipocytes, some fat cells. So what happens is we bring oxygenated blood, fully oxygenated blood into those tissues, but on the other side of it, we are bringing fully oxygenated blood away from the lungs. And conversely, we are bringing lesser oxygenated blood, what we would color blue, because it has so little oxygen in it, back to the alveoli. But the reason why that blood is blue in the first place is because at the level of the tissues, it became deprived of oxygen. Now let's follow the numbers. That becomes the more confusing part of it. First of all, we want to understand that the lumen of the alveolus, or the interstitial fluid surrounding those tissues, is going to be what sort of drives the numbers and affects the blood in the first place. So if you look at the lumen of the alveolus, it begins at the lumen of the alveolus will have 40 millimeters of mercury of carbon dioxide and 104 millimeters of mercury of oxygen. And when we look at the interstitial fluid surrounding the tissues, the carbon dioxide levels are greater than 45 millimeters of mercury and the oxygen levels are less than 40. So how did these numbers come about? Well, if you look at the alveolus in the first place, the alveolus is basically the end of a tube, a very long convoluted tube that is open to the atmosphere. And the atmosphere numbers are quite different from this, but the reality is that a certain amount of air is always left behind, this residual volume that is left behind in the lungs. And so you're always going to have a much higher level of carbon dioxide in the alveoli and a much lower level of oxygen in the alveoli as compared with the outside air. And if you look at the tissues, where those numbers come from is the idea that's, this is kind of a logical, you have to kind of separate yourself out from the logic of the situation for a moment, or you have to really embrace it. And the understanding here is that if you have cells that are metabolizing, that are undergoing aerobic respiration, well, the cells themselves really need oxygen, so they're constantly sucking it up. As soon as there's a little bit of oxygen in the interstitial fluid, the cells are going to be grabbing it and using it up, and of course outputting carbon dioxide as a result. So in the interstitial fluid surrounding tissues that are metabolizing, that are, they're demanding of vascularity because they are, it's a muscle contracting, or it is a kidney cell that is pumping, sodium ions or something like that. In those cases, you have to have a good oxygen supply, but as soon as you have that oxygen supply, the cells take it up. So it leaves the interstitial fluid deprived of oxygen and rich in carbon dioxide, relatively speaking. I think once you kind of have those rules established for why the lumen of the alveolus looks like that and why the interstitial fluid looks like this, it's a little bit easier to understand where all the numbers actually come from. So if we just follow the carbon dioxide story, well, it leaves the tissues at 45 millimeters of mercury and it enters the alveolus at the same number, at 45. And then as the blood makes its way, and we're going to take a closer look at this, but as it surrounds the alveolus, it's going to perfectly exchange through diffusion and equalize the numbers. It's going to equalize the blood with the lumen of the alveolus, and so the lumen of the alveolus was 40 millimeters of mercury, so the blood then becomes 40. And then if you follow that all the way down to the tissues of the body, of course, it's still 40 as it approaches the tissues, and as it equalizes out with the surrounding interstitial fluid, of course, we said that that is rich in carbon dioxide there because the cells are respiring and constantly releasing carbon dioxide. And therefore, the blood equalizes with that interstitial fluid and goes back up to 45. Now, in between here, I did not actually write the heart, but you should recognize, of course, that the heart is in the center of all this. I kind of feel like it's not super important to actually draw the heart, but understand that, of course, blood passes through the heart, which allows it to be pumped through the arteries, through the pulmonary system to the lungs, and then through the systemic circulatory system to the tissues of the body. I think we covered that enough, but so I didn't really feel the need to put that detail in here. I thought it just added to the story, kind of unnecessarily, actually, because the heart doesn't actually have anything to do with either adding oxygen to or taking carbon dioxide out of the blood. It's just the lungs that do that. So we are not doing the heart in this particular story. Right, and so let's add in those oxygen numbers. As the blood leaves out of the tissues, it's colored blue because it has a partial pressure of oxygen of 40, and that stays true until you get all the way back to the lungs, and it's going to be 40 as it enters those lungs. And of course, we diffuse and equalize as we pass through the alveolus or pass next to the alveolus, and then the blood becomes 104. It becomes more oxygen rich. And that almost entirely stays true all the way to the tissues. The reality is that not every single alveolus is completely exchanging everything properly. So by the time we get to the tissues, the blood, the partial pressure of oxygen in the blood goes down from 104 to 100. It's a small difference, but it just means that it's not every aspect of the lungs are completely efficient and exchanging perfectly. So if you look at one alveolus that's exchanging perfectly, it will be 104, but the lungs as a whole are at 100. This oxygen-rich blood is colored red at this point. It approaches the tissues and, of course, exchanges and diffuses and equalizes with the interstitial fluid, which you recall, or less than 40, because the tissues are always taking up oxygen and leaving the interstitial fluid deprived of oxygen. And so that becomes the new number for the blood, 40 millimeters of mercury. I wanted to do something a little new here where I simply drew an entire alveolar sac, and I wanted to draw how blood actually surrounds this. I wanted to show the depth and richness of the vascularity of an alveolar sac. So we draw it kind of like a bunch of grapes. We have blood that comes in, which is oxygen deprived, and every single one of those alveoli is going to have its own set of capillary beds. We're actually only drawing one, but there will be multiple, probably. 80% of the surface of the alveoli is covered with blood vessels. It's an extraordinarily vascular situation here. And so we'll notice, if we pair this with the prior lesson, what that means is that every single one of those alveoli inside of an alveolar sac is doing that level of exchange at the same time, going from 40, the blood is going from 40 millimeters of mercury to 104 millimeters of mercury, all at the same time. So I just wanted to give that complexity to the situation. And now we're going to look at one single alveolus very, very close up. Yes, we are just pounding home this idea of the external respiration, because I do feel it's extremely important to understand this properly. So the alveolar sac is made of simple squamous epithelium, which we are drawing in black, flattened, squashed cells, each of which are alive, each of which have their own nuclei. And then surrounding it, very tightly bound to it, is going to be one wall of a blood vessel, a capillary, which is also simple squamous epithelium. And you notice that about a third of it is blue and 2/3 of it is red. We'll come back to that. These two layers together are called the respiratory membrane. It's exceptionally delicate and thin, and no wonder it gets damaged so easily, like, for example, with COPD disorders, with emphysema. It's just extraordinary. It's actually literally fragile. These two cells together, these two cell layers together, are 1/15 the thickness of tissue paper. So it means that, yes, things can be easily exchanged in and out. Oxygen is able to easily cross through the respiratory membrane and enter into the blood supply. and then carbon dioxide is also easily exchanged out of the blood supply and into the lumen of that alveolus. Well, keep in mind, of course, that this is a blood vessel, so it's going to have two walls. The other wall is also made of simple squamous epithelium. Again, about a third of the way around is going to be blue, and then 2/3 of the way around is going to be red. And just to drive home the idea that what the blue and red mean is that the blood starts off, and we'll draw these arrows going through there. It starts off blue and it becomes red, starts off at a lower oxygen level, say 40, and becomes a higher oxygen level, say 104. And the switch is made at 0.25 seconds. Let's go ahead and do an XY axis where we're going to discuss the timing of external respiration. How long does it actually take under healthy conditions when you are at rest? How long does it take for full oxygenation to take place? Well, according to our little drawing here, it's 0.25 seconds. So on the x-axis, we're going to do time in seconds, and on the y-axis, we'll do the partial pressure of oxygen. We're going to be measuring the level of oxygen in the blood as it moves its way around this capillary bed, around this alveolar sac. And the three time points on the x-axis are going to be 0.25, 0.5, and 0.75. These are significant. So we go from our lowest level, which remember is 40, all the way up to 104, and it takes about 0.25 seconds, 1/4 of a second, 1/4 of a second to make its way to the point where it is actually fully oxygenated, which isn't very long, and it turns out it has a full 0.75 seconds to actually do that. And you can imagine the way that this could change if, for example, you were exercising and the blood was coursing through your body faster. Your blood velocity simply speeds up, and therefore it's also going to speed up going through these capillary beds. And in that case, then what will happen is you will not quite have the 0.75 seconds. You might have 0.65 seconds. You might have 0.6 or 0.5, depending on how fast your blood is traveling. But nonetheless, you still only need 0.25 seconds in order to have full oxygenation. So as long as you are still exchanging properly and everything's healthy, that's still enough time, as long as it's more than 0.25 seconds. And as long as your lungs are actually healthy. So what we're actually going to explore in this curve is we're going to add in a variable where your lungs are not healthy. Remember emphysema, where your alveoli are actually breaking down? What will happen there is that your exchange is not working very well because you don't have enough surface area for those cells are not very healthy, and therefore they're not allowing for full exchange at quite the fast rate as before. And therefore, it might take a little bit longer for the actual exchange to happen. So rather than full oxygenation happening at 0.25 seconds, it might happen at 0.35, 0.45, 0.5, or even longer. This then doesn't give you many options for example, exercising. You no longer have the ability to exercise. First of all, of course, your muscles are working away at this whole business. We alluded to that earlier. But the other side of it is that you simply don't have enough time for there to be any kind of differences in the time course for that blood to make its way all the way around the alveolus. It becomes quite dangerous, of course, if it takes you 0.6 seconds to fully exchange oxygen and fully oxygenate your blood. You really don't have very much buffer between that and 0.75 seconds, which is the amount of time that normally it would take. So it would be very difficult for you to exercise, I guess is what I'm saying, if you had especially end-stage emphysema or emphysema that was fairly advanced. You couldn't. There isn't a lot of flexibility there in this system. Well, as you can imagine, and as you probably kind of just know anyway, the lungs are one of those places where you are most likely to get infected by different viruses and different bacteria, because it is, it's this open tube to the air. So whatever's in the air, you don't have a lot of protection. Of course, you have a mucous membrane, you have the mucosa-associated lymphatic tissue, but other than that, it's basically a lumen, this tube that goes through, the pharynx, larynx, glottis, trachea, bronchi, bronchioles, and all the way down to the alveoli. It's open to the air. So keep in mind that the lumen of the alveolus is actually outside of the body, because it is open to the air. So that keep that in mind, that when you are exchanging air into the blood across the respiratory membrane, across the wall of the alveolus and across the wall of that blood vessel, that is air entering the body. When oxygen moves through that respiratory membrane, it's going from the lumen, which is the outside of the body, from the lumen of the alveolus, which is the outside of the body, to the lumen of the blood vessel, which is inside of the body. This is one of the most vulnerable places in your body. And this is oftentimes where you feel very sick from, like a rhinovirus, a cold, a coronavirus. And so we have lots of protections. Not only do we have all those mucosa set up on the, as the air journeys through, and oftentimes, to be honest, that mucosa does a really good job trapping illnesses in like the upper respiratory system to ensure that it doesn't make its way into the lungs. But of course, sometimes things do make their way all the way into the alveoli, and what we have then crawling around outside of your body in the lumen of the alveolus are macrophages, and there's a lot of them. In fact, there's so many macrophages making their way. You might recall that the macrophages are a really powerful part of your immune system. They allow for the secretion of cytokines, for example, that can enhance the inflammatory response very quickly, which is why, again, it's a vulnerable place in your body for that other reason too, because they can enhance your inflammatory response, and sometimes your body really suffers as a result of that. It can lead to pneumonia, for example, bringing fluids into this region. But anyway, we have lots of macrophages there. The purpose of the macrophages is, of course, to detect any kind of foreign matter coming along, bacteria, viruses that happen to be approaching this very delicate tissue in this region. And in fact, there are so many macrophages that you actually cough up and then swallow back down, like not cough up, but you actually, like 2 million of them per hour make their way up your mucous membrane into your pharynx, and then you actually swallow them down the esophagus. 2 million per hour. So there's a lot of them. For our last big topic today, we're going to be discussing the respiratory centers, and in fact, we'll be discussing 3 different aspects of the respiratory centers. I find that students really struggle to actually separate these out, so I'm going to be trying to be very clear about what's happening here. The inputs to the respiratory centers will be in other parts of the body. Chemoreceptors are found in the aorta. They're going to be detecting things like carbon dioxide levels and acid levels. We'll be talking about those more actually in the next lecture. Pulmonary irritant receptors cause you to cough when you have irritants come into your lungs. And baroreceptors will not allow for the lungs to hyperinflate. In case you've breathed in quite a lot of air and you've inhaled quite a lot of air, the baroreceptors will not allow you to damage your alveoli by overinflating. The root baro means pressure, so they're pressure sensors. So both the pulmonary irritant receptors and the baroreceptors are found in the lungs. And then there are higher brain centers. So the respiratory centers are going to be found in the brainstem. But there's other parts of the brain that are responsible for also helping to contribute signals to the respiratory centers. The hypothalamus would be, for example, responsible for, for example, it's responsible for body temperature regulation. So when you unexpectedly plunge into a cold region, you tend to gasp. So the hypothalamus is feeding into the respiratory centers in that way. And of course, your cerebral cortex, which is responsible for your voluntary control, for example, over movement, would allow you to do things with your breathing rate that would be outside of the involuntary physiological function that they normally are. So although your brain centers for respiration are involuntary, you can use your cerebral cortex to make choices about what you want to do. For example, you can undergo apnea, which is holding your breath. And that would be a choice that you make. You decide to stop breathing for a little while by inhibiting those respiratory centers. So all 5 of these inputs, chemoreceptors in the aorta, pulmonary irritant receptors and baroreceptors in the lungs, and then the hypothalamus and the cortex, which are other parts of the brain, all 5 of these, and probably other things as well, will feed directly into the brainstem into your respiratory centers in order to either speed up your breathing rate, for example, or slow it down depending on what is needed, or cause you to cough, for example, or cause you to gasp. All of these things would have that input. But when you're actually looking at the part of your brain that's responsible for controlling your respiration, that would be more directly these three parts of the brain that we're drawing now. So the inferior-most part of your brain, of course, is your brainstem. We have the spinal cord down below. We have the pons and the medulla, which are kind of subtle outswellings of the brainstem. The pons is going to have one of the respiratory centers, and the medulla is going to have two. So the one in the pons is called the PRG, the pontine respiratory group. And then the one that is posterior in the medulla is called the DRG, the dorsal respiratory group. And the one anterior in the medulla is called the VRG, the ventral respiratory group. We actually used to think that the DRG was the pace setter for breathing, but now we have discovered that it is more likely the ventral respiratory group that is the pace setter for breathing, which allows you to have a eupnea, or a true breathing rate, of about 12 to 20 times per minute, at least when you're resting. So what happens then is that the cells of the ventral respiratory group will depolarize 12 to 20 times per minute, and I'll show you what they do in a moment. Now, if we take a look at the role of the PRG, the pontine respiratory group is basically responsible for the transition between inhalation and exhalation. So most of the time this is considered to be an inhibitory connection because it's causing these different changes. So we're transitioning from inhalation to exhalation. This is considered an inhibitory connection. So we put a negative sign by those arrows there. And as far as we know, the PRG does actually have a direct connection directly to the DRG, but the DRG has input directly to the VRG. So the dorsal respiratory group is considered to enhance the activity of the VRG. So we draw a little arrow there from the DRG to the VRG, and we put a little plus sign there to indicate excitation or enhancement of the activity. So most models of this, and I think we're still kind of working out exactly what the respiratory centers actually do, but most models will have the input going directly from the VRG to the muscles of inspiration. And there are actually a few different muscles in your thoracic region that are involved in respiration. But the two biggies, the two main prime movers of respiration, are going to be these muscles that strap on the external surface and between the ribs, which is the external intercostal muscles. And then, of course, the much better known one is going to be the flat dome-like muscle at the bottom of the thoracic cage, which is called the diaphragm. So remember that I said that the cells of the VRG, the neurons of the VRG, will depolarize 12 to 20 times per minute. And what they do when they depolarize is they cause contractions of these muscles, which in turn causes you to inhale. And then when the VRG is relaxed or quiescent, the cells are no longer active, the muscles will relax, which causes you to exhale. And I thought that I would just mention Boyle's law at the very end here, because we just can't leave it like that. We've kind of left a big hole in the information by just saying, oh, contraction to these muscles causes inhalation, and relaxation of these muscles causes exhalation. This is to do with Boyle's law, which there's a few different ways to write Boyle's law, but in my class I always just teach it as PV equals K, K being a constant. And what this means is that P equals K over V, or V equals K over P, which means that P and V are inversely proportional to each other. So if you just look at that and you understand that P means the pressure of the lungs, and V equals the volume of the lungs. What that means then, remembering that these are inversely proportional to each other, as the volume goes down, the pressure goes up. As the volume goes up, the pressure goes down, because they're inversely proportional to each other. So if we kind of have a little bit of a grasp, and you don't have to understand this perfectly, but if you kind of have a grasp of how that works, then all we have to do is understand what causes the volume to go up or down, and then at the end of the story, what the pressure going up or down means. So the way that the volume of the lungs goes up is that we contract these muscles. When you contract the external intercostal muscles, it causes your rib cage to pull up and out. As you contract the diaphragm, it causes the diaphragm to pull down. And this in turn increases the volume. And then when these muscles relax, of course, the opposite happens. As the external intercostal muscles relax, the rib cage pulls back in and down. The diaphragm, when it relaxes, it actually domes back upward, it curves back upward. And so we increase the volume of the thoracic cavity, the thoracic cage, by contracting both of these big old muscles at the same time. And we decrease the volume of the thoracic cage by relaxing them. Well, so if we are going back to Boyle's law, as we increase the volume, the pressure goes down. Now, pressure going down creates a vacuum into which air will rush if it can, which of course it can in this case, because remember that the alveoli are open through this long series of tubes to the air. So as the pressure inside of your lungs goes down, it actually pulls air in. So as the volume increases, the pressure goes down, creates a vacuum, air rushes in, and this is how you inspire air or inhale air into your lungs, into your alveoli. And the opposite is true. As those muscles relax, the volume decreases, the pressure goes up, and this pushes the air out, and you expire the air or you exhale the air.